looking forward to the pizza.
Okay.
And I'm going to tell you a little bit about a couple of projects we've done using Delta Maps, which is a machine learning tool that we developed in 2016-18 with Konstantin Dobrolis. At the time, he was a professor in the College of Computing. Now he's back in and a student in the College of Computing, a graduate student, Ilias Notalis. And here we go. So what is the problem that we're trying to address? If you want, first of all, we are seeing a lot of changes in the marine ecosystem associated with anthropogenic changes. So generally warming and also acidification, but it's really warming the main driver. We are seeing new species getting into places where they didn't used to get, and that's a problem. And especially is a problem in the Mediterranean Sea, where there are now invasive species that did not exist until 10 years ago that are really killing everything else. And the Mediterranean Sea had already a lot of problems due to overfishing. So having those invasive species, it's overly problematic because the system was just trying to recover after we start implementing rules about when and how much you can fish to try to revamp a little bit the ecosystem.
Recording in progress.
Having those arrivals of invasive species, and I will show you what I mean by that, has been problematic. The other one is corals. And corals are foundational to the marine ecosystem. So coral reef, surface coral reefs are the base for pretty much every tropical fish ecosystem. What I mean here is that if you kill the corals, you kill the whole food chain above them. So you get rid of every single fish that live because the coral system exists. In some areas, especially in the Atlantic, we have lost 75% of coral coverage. They're dead, period. In other areas, they're doing a little bit better, especially in the Pacific. And I will try to show you that we may try to understand why using this system. But if you want to understand what's going on in the marine ecosystem, you have to manage to figure it out. You have to map the ecoregions. And the ecoregions are simply regions where you have relatively homogeneous areas in the ecosystem. So you have more or less the same species distribution, and the same conditions. Similar temperatures, similar nutrient like nitrogen and phosphorus supply and so on. And the reason you want to do that is that if you have to have any kind of management solution for species conservation, you have to know what you are preserving and how. And so you have to kind of map the distribution of what you have and where they are similar or not. There are 4 ways to do that. One is taxonomic, one is ecological, one essentially integrates the taxo-ecological ones. Those 3 relies on in situ observations. The ecological is easier because it's simply going and measuring biogeochemical and physical quantities. So you put an instrument in and decide what is the average temperature here. And because we're looking at near surface system, let's say after 100 meters, you can actually use satellites to at least have a map, overall map of temperatures. The taxonomic is the one that really goes in and looks for species distribution and is expensive and is very time consuming because what it means here is that you have to send a scuba diver that take in account what you find, right? So it tells you those are all the species we have. The last one, it's connectivity based and essentially looks at the impact of oceanic currents on larva dispersal. And so figure it out how things may be related by physics, because at the end of the game, we know at this point from genetic analysis that physics seems to be key in how things get distributed. The last one is the only one that you can get rid of the in situ observations, at least in part, because you can think of doing it with models. And there has been a lot of effort, especially since Lisa Levin from Scripps, really famous oceanographers there, like 10 years ago came out saying we really need to map connectivity because that's the only way we can put forward measures to try to preserve some of those ecosystems. And the reason it's very simple. If you have a system and that system is a little bit endangered for whichever reason, if that system can receive a bunch of larvae from similar ecosystems further away, that system has the opportunity to rebuild itself because it gets fresher genetic material from another place. This is especially true for corals. Corals can bleach and can look really in bad shape, but you do maintain the coral structure for quite a big long time. And so if new larvae from corals from other reefs that have not been bleached can come through and populate that coral, they can rebuild the population really quickly. And that coral will not die. Okay? So we want to know that. So what we really want to see is, how propagles, just tiny fish and larvae disperse. And that is going to tell us how the adult organisms are going to be distributed. And this is a really good review that was published in 2006, so actually it's 20 years ago. That really, put the community, raise the community awareness of the fact that we need to understand larva dispersals if we want to try to do anything that matters in terms of management. And this caused a lot of effort using modeling to try to do that. Unfortunately, it's very computationally expensive. And so you can do it for a few years, essentially, of time. The problem is that we are changing the system, and the climate system per se, the current system in the ocean, does change on multi-year up to decadal timescales. And so just with modeling is very hard to do it at the scale that you need. So essentially, we need a system that it's able to inform us about connectivity at basin scales and on time scale that are of the order of several decades. We propose an approach called Delta Map that initially was not at all thought for this kind of application, was thought and was developed to identify teleconnections in the atmosphere. Teleconnections is essentially how the atmosphere transfer through Rossby waves and Kelvin waves information about changes in winds and changes in atmospheric patterns from one region to the next. So you can identify teleconnections looking at sea surface height, sea surface temperature, winds, geopotential height in the atmosphere. And so what we wanted was to develop a system that would allow us to look into models or the analysis and just identify the patterns without starting from indices, which is how scientists used to do it. So you would define an index, for example, the El Niño 3.4 index in the Pacific, which is as a sea surface temperature anomaly in the Pacific, and then do a regression on that index, or do a EOF, and look at the patterns. The problem is that EOF have a problem of being always perpendicular, and so you don't know if what you actually obtain is really independent or not. Okay, well, if we can do it with a machine learning system, then it's going to be easier. We'll not have the problem of this requirement of being perpendicular in the EOF, and also will not depend on the definition of the index, because the index may work in the observation, but may not work in the climate model may not have the center of interactions exactly where are in the observation. And so if you're defining an index based on the observation, it may not work when you go on the other side. We found out that if we apply the same machine learning tool that I will describe next on sea surface temperature anomalies, okay? So we subtract the mean over multiple years, and we do subtract the mean every month. Okay, so we subtract the seasonal cycle, we just look at the anomalies. Those anomalies are very, very strongly correlated with the sea surface height, which is essentially the height of the ocean, and it's a measure of the geostrophic currents. Okay, so what the SST, think about the ocean surface. You have two forcings. One comes from the atmosphere, and one is intrinsic to the ocean because you have currents. Okay. Now, the atmospheric signal between, let's say, 40 north and 40 south is very large scale because the Rossby deformation radius of the atmosphere is much larger than the Rossby deformation radius of the ocean. So the heat fluxes will determine what your SST are, okay? Because you are warming or cooling from the top, and the ocean is responding to that. Currents are going to determine variability at scale that are a little bit smaller than what the atmosphere can do. Because the Rossby deformation radius is smaller, and your variability in terms of current is associated with the stability that you have in the ocean, which depend on that Rossby deformation. So your sea surface temperature anomaly, month by month, depend on the variability of the currents, not on the variability of the atmosphere. If you look at scale that are small enough and that are close to the Rossby deformation radius of the ocean. In other words, you can use the sea surface temperature anomaly as a proxy for currents. And this has been shown in the previous paper in 2001 by comparing that sea surface height and geostrophic velocity derived from sea surface height observation from satellites and sea surface temperature. So at spatial scale of 10 kilometers and frequencies of month, even work in summer, or people that have worked a lot on satellites, show that essentially you have a very strong link between the anomalies that you have in the temperature and what your currents are doing. So essentially we can use the sea surface temperature anomalies to determine connectivity currents. That's the intuition that was in this project. Then we use this unsupervised learning mechanism, which is a complex network tool that identifies contiguous and possibly overlapping domains. And a domain is especially contiguous region where all the grid cells participate in the same dynamic function. So they have a very highly correlated temporal activity. And then we can define a network across those domains that essentially tell us the magnitude of interactions between different domains. And then we can define the strength of the domain depending on how many interactions it has and how strong. And so we can also measure which domain is really key in talking to the others versus receiving. I'm going to show you the first application to the Mediterranean Sea of this idea. So we had this tool done for different reasons, and then we had this intuition, and it was really by chance that applying it to sea surface temperature anomalies, just because I tried, I was on sabbatical, I don't know what I was doing, and I kind of tried, and then I talked to a marine ecologist who told me that the map I was showing him in the Mediterranean Sea really resembled his map of connectivity from actual observations. That's how this idea came up. So this is the Mediterranean Sea, and we had a very big problem from invasive species from the Red Sea that come through the Suez Canal. Okay, and the Mediterranean Sea is now full of lionfish, and lionfish cannot be eaten by anything and destroy a lot of stuff that is smaller than it. And so it's really decimating the marine ecosystem that we have in the map. This is another issue that we have to consider when we're looking at the Mediterranean Sea. This is a trend in sea surface temperature for the last 30 years. The Mediterranean Sea has warmed a lot. Okay, this is per year. So you have to multiply by 30 in order to get the total. has not warmed uniformly. The east has warmed much more than the west. And this has been a problem for the lionfish because the lionfish really loves high temperatures. Okay. And this is a reanalysis of sea surface temperature data, which has a 4-kilometer resolution. So very good resolution for getting all the currents in the right place. Okay, so it's assimilating all the possible measurements that we have, and is giving me.
So how is computed that map?
That map is just a linear trend from the reanalysis. So I'm just taking the temperature, the sea surface temperatures are from satellites. The reanalysis is putting them at 4 kilometers, but is assimilating all the satellite information. And so I get the map and I take just a linear trend. It's quite linear, so it's not a bad approximation. We'll check that, but you know, it's not perfect, but still gives you an idea of how much has warmed up. This is a work that came out in 2014, and that did what I told you it was done before. So you take a model, you run it, you have the currents, and you put Lagrangian particles, and you see where the Lagrangian particles go. And you put Lagrangian particles continuously. And then you define those ecoregions based on where the Lagrangian particles stay. Okay.
They are on the surface.
Yes, they are in the upper 100 meters. Okay. And that's just because fish and larvae, etc., pretty much live in that area. And so this is the eco-regionalization that they came up with. And this was a 3-year period. And to do all the experiments, so both the Eulerian field and then the particles, because you have to save a lot of data. You have to save it very, very frequently to do it at a good resolution. Okay, so first run the model, you save all the output of your model, the velocities, and then you put the particles in it. This project took about 2 years. Now, you can do it faster today because we have better computers, but nonetheless, it's a very long process. So you can do few years, you can maybe, today you can do 10 years, but you're not going to do much more than that. It took us less than a day to get pretty much to the same ecoregions by using delta maps. They're not identical, but they're not identical also on the velocity field because we're using a reanalysis, they just run a model. Okay, so our 2 different baselines to begin with, slightly different for the same reason. But it turns out that we actually get, if you map this on the previous one, you pretty much get the same ecoregions. The advantage of this is that we also have automatically from the strength, and this is a strength map, an idea of what are the regions that are very strongly connected and which are not. And it turns out that we map very, very well in that regard, what that data said. So wherever you see red, those regions are very strongly connected, what they have around. Whenever you see blue, they are quite isolated. So stuff stays within the regions, but doesn't really move into the other ones. Okay, there are very weak connections between regions that have a very low strength, and there are much stronger connections between regions that have high strength. So for example, this area is very well connected. This area is very well connected in those 3 years. Okay, here we did exactly the same 3 years, just to compare and to have a validation. And we also showed, for example, that there is a very strong correlation between the sea surface anomalies and the velocity field all the way to 150 meters. So we're sure that what we're doing makes sense. Then we look at 30 years, that's what we had. And there is where you must be a climate scientist and have an idea of what you're doing. Because of course, over 30 years, there must be some kind of variability that is determining how that system works. And for every basin is a little bit different. In the Mediterranean Sea, a very strong signal comes from the kinetic energy. So you have periods where the kinetic energy is very high and periods where the kinetic energy just goes down or up. Okay, so those are pretty significant differences in kinetic energy. And so when we then had to do all this analysis, we picked 3 periods One where the kinetic energy is growing, one where the kinetic energy is more or less flat, it's kind of varies around, and one where the kinetic energy is coming down. And then we run delta maps every 3 years, proposing 1 year at the end. So we did many runs. And then group them, checking that it makes sense to group them in each of those periods. So actually, no, we did it in time slot of 4 to 9 years. And then we assessed the system in each of these 3 sub-periods, and then we found that the best grouping was 7 years. That was the best trade-off between using single slot of equal lengths and whatever. But
why is the kinetic energy negative?
Why is the kinetic energy
Negative.
Oh, this is just a trend. So there is this is on top of the baseline.
Oh, okay.
Kinetic energy anomaly, if you want.
Oh, okay.
In oceanography, we have what is called eddy kinetic energy, and so here we had to be distinct from that, because in reality, what we did is really just taking the trend over those periods. So you have an average kinetic energy that has been subtracted from here.
Oh, okay.
And so we find the ecoregions for those 3 periods. And essentially, we also find that there is that the number of ecoregions is very different in the 3 periods. And that there is a very strong correlations between what happened, especially in the Eastern Basin, and what the energy was. Okay, and I'll show you how that changes. So this is the situation. So this is the first period where the energy was going up. This is the second period where the energy was kind of stable. And this is the third period where the energy is coming down and is going back to what it was initially. The interesting thing of this plot is that it clearly shows that most of the changes are in the Eastern Med, right? Because the West remains relatively well connected all the time. But the East is really going from periods where things are connected to periods where things are not connected at all. The interesting thing of this is that here we have, in this time, we have the first observations of lionfish emerging also around the coast of Lebanon and Turkey. But they were very isolated, and the temperature at this time in summer, very rarely, the surface temperature reached 28 degrees Celsius. Lionfish to larvae to grow, lionfish eggs to mature need more than 28 degrees water. So we had lionfish coming through at this time. It's just that it could not make more lionfish. So there were single fish that came in through Suez, but didn't find the right environment to reproduce. Temperature definitely got warmer here. At this time, we had already quite a bit more than 28 degrees Celsius water. But everything, it's not connected and really destructures in this area. So essentially, the lionfish was limited to stay where it was, which is around the Suez Canal. Could not get around the basin. Then in 2011, things started to change, and we started having a very well-connected area here. And in fact, we started in 2012 to see observations of lionfish all around these areas. And then from here, it went here, and we have observation here, and then we have observation here in 2016. And in 2017, we started having observation in Sicily. So we actually managed to map the propagation of those lionfish. And the population grew incredibly because at this point, the waters are absolutely fine for them to reproduce. And So we've been able to follow essentially the propagation of the lionfish. I was quite surprised because we really match really nicely the observations, and I was not necessarily expecting that. So this is essentially the story that I just told you. And what it's linked to, it's a change in the circulation in the east, which is known. There is a mode of variability in the Eastern Mediterranean Sea that has a decadal time scale where really the circulation changes dramatically. And so the water that comes from here turns up here and then recirculate back down or goes all the way around. Okay, so we really know about those two patterns. And essentially in this period, the water was going up, and in fact, you can see that there is all this linkage here, and we are mapping it and then coming back, and here it was isolated. And in this period, we're back where we are doing this whole tour. And we can follow that the velocity. So this was essentially the first time that someone did a study of connectivity over 30 years. We're very happy because we could explain a lot of the data that there are, which are very sparse, because they're essentially fishermen reporting the first time they see the lionfish, and then you follow in the fishery publications how that distribution evolves. We decided to try to look at another big question, which is why the Pacific is doing so well in terms of corals. And what I mean here is that, again, in the Atlantic, we lost 75% of corals. In the Pacific, we didn't. They get bleached and they tend to recover, not everywhere, but we do know of reefs that have recovered 100% of their coverage within 2-3 years from a big bleaching event. And so the question is why? And I can tell you that we also tried in the Atlantic. We still haven't managed to publish that, but it's under review. And what I'm going to tell you today, which is essentially because the connectivity is very, very strong here and very, very variable, makes sense also with the Atlantic, because the Atlantic doesn't have those characteristics. So this is the coral triangle, and this is the most diverse marine ecosystem we have in the world. This is the distribution of reefs. And we're going to consider a broader area than just the coral triangles. The coral triangles is here. And where you see the blue dots, so every single red dot is a coral reef. Where you see blue dots are the locations where we have that kind of taxonomic observations that would allow eventually to do taxonomic connectivity. So very, very few. Those are just the reefs for which we have enough to say which species exactly we have. For the other ones, we just don't have those data. 75% of reef-building coral species live here, 40% of non-coral fish, and 75% of mollusks all live here. It occupies 1.5% of the world ocean, but actually has 30% of the world reef area. And has been facing a warming that has varied between, in terms of minimum and maximum, between 0.09 and 0.12 degrees C per decade, in terms of minimum and maximum temperature changes. And the projection is that they are going to climb between 1 and 4 degrees by 2100. So this is a big biodiversity hotspot. I'm pretty sure that everybody has listened to the news at some point for the Great Barrier Reefs and all the bleaching that occur here. Spoiler alert, from our work, the Great Barrier Reef is doomed. It's not going to recover. And in fact, it's one where we are really seeing not a lot of recovery. So we did exactly the same thing, but we added a piece. We managed to rank the biodiversity through a centrality score. And what the centrality score is, this was really good idea of the postdoc that worked on this project, Luba Novi. She essentially used what Google has developed to measure the popularity of web pages. And so she's saying a region is popular if it has a lot of connection, if it's receiving a lot of potential larvae from a lot of places or from places that really release a lot. And then we also managed to put together the information that we have about bleaching. And so identify reefs that are high risk of dying or that have a high potential of recovery. And so actually make maps of that and then figure it out why. And this becomes really important because if you can do that, then you really know where you can focus your effort in terms of marine protected area and actions to take to try to save those reefs. So we are in the tropical Pacific, we are in the Coral Triangle. There is one key climate player, and that is the El Niño Southern Oscillation. Okay? That's the most important mechanism of variability for sea surface temperature there. And what I'm showing you here are maps of bleaching, essentially, of alert chance of bleaching that NOAA releases every week about corals. And what you can see, this was actual September last year because we have a big El Niño, so we were all worried about the corals. But 2022, so this was El Niño. So this was El Niño map, and this is an El Niño map from 2015. Now I could put the one from last year better. What you can see here is that the red, which is where you really are losing corals, okay? Where is red that their corals are dying? The red patches are very different in those two maps. So a strong La Nina versus a strong El Nino are going to really do damage on different areas. The other thing that you see is that through the last 30 years, we had a bunch of very strong El Niños and a bunch of very strong La Niñas. And you can divide the years pretty much in 3 parts of equal length. Neutral, El Niño, or La Niña, basically with that characterization. So we did that, and then we looked at the ecoregions and their strength. And this is what comes out, and this is really interesting, because what this is showing you is that the strength and the regions are very different depending on which El Niño condition you are. And so condition you are. So this is for El Niño, that's for La Nina. Everything is very, very connected in the middle. And there is a very strong connection between the Pacific and the Indian Ocean during La Nina. You don't have that at all in neutral years. And you have much less during La Nina. Okay. What this is essentially telling you, because remember, the strength is telling you how much exchanges we can have, is that you have completely different larvae that comes to the same place, depending on which period of El Niño events you are. And so essentially, the same reef, depending on the year, is going to receive larvae from very different places. So one year we receive them from the left, and maybe the next year we receive them from the southeast. And that helps, because usually you don't bleach everything at the same time. And so within a year or 2 is going to get new larvae that were not bleached originally. Because this is an interannual variability that every 2 years or so you're changing, right? So essentially, a lot of reefs can manage to receive fresh, healthy larvae from many different locations. It's very dynamic, it's not a static system. You cannot just do eco-regionalization once and make that picture to be static. Every year is going to be very different from this one. And that helps. So you can also group them together and really do community analysis. This is something that if you're doing machine learning, you really like, and you can really see even in the community analysis that you have very different communities that show up. So those are communities. What this means is that you have a very large chance to exchanging genetic material where you have the same coral. So here you have this group that is probably going to be able to exchange material. Here you have this group during La Nina years, those two groups are going to be very separate. Which also means that if you can save one of those two during La Nina year, right, you are going to essentially, for example, if La Nina does not matter for the Indian Ocean, which is the case, the corals here can exchange larvae and send them here and populate this area that maybe is being bleached. And the same is true for the other, and you can see that there is really a different distribution of those communities. And that is really what is making the Pacific to be able to do much better than the Atlantic is. In the Atlantic, that's not the case at all. The communities are always the same. And so if you manage to bleach the link between two weakly linked communities, you're done. And then if you bleach, then one of the community is not going to be able to regrow. And so here is where I explain the page rank centrality. So the idea that you are popular in terms of coral reef if you can receive a lot of larvae, or if you receive larvae from few reefs, but reefs that are very biodiverse, which means that you're still receiving a lot of different larvae. So we make a biodiversity map based on that, and this is what happens based on the regions that we had. And so you can see that also the areas that are popular, what it means, the areas that can receive many different larvae are very different in different periods of El Niño. Okay. So for example, in this period, during La Nina years, this region gets a lot of different larvae, and this region gets quite a few different larvae. During El Niño, this region really doesn't get anything of diversity, but it gets them here. You want to receive larvae as much as possible from different places, because that is going to increase the chance of recovery. And, you know, a different map is a neutral here. So overall, this system is incredibly dynamic because of El Niño. And it's interesting because ENSO is being considered the main cause for bleaching in this area, because ENSO is so important in terms of bleaching events. What we discover is that really the biodiversity of that region, it's due to ENSO. So ENSO, first and foremost, is what created this hotspot in biodiversity. If we didn't have ENSO, we would not have the diversity of reefs and fish we have in that area, because it's really helping the exchange of larvae from one place to the other, and the survival of those places by being so dynamic. So now that we have all of that information, that we have the NOAA maps for the past 30 years of who got bleached and who didn't, we can put all this together and build an index that calculates, take into account for the risk of bleaching, because we have the bleaching maps for the last 30 years. So if a place was bleached once over 30 years, the risk of bleaching is low. If it was bleached 5 times over 30 years, the risk of bleaching is high. And we can make an index of that. And
What is bleaching?
oh, I'm sorry. When a coral is, so a coral is a symbiotic organism where you have the coral and you have an algae that lives in the coral. And that algae is what gives the coral the color. All corals are white, surface coral per se. It's the algae, Solventella, that gives the coral. If the water temperature is too high, an algae, which is a plant, doesn't like it, leaves the coral. The coral, it's white.
Is a
Or
good
it's
thing
left
or
white.
bad things?
And that's called bleaching.
Is a good thing or
No,
is bad?
it's really bad. The larvae, the algae is what collects nutrients for the coral. So the coral without the algae will die. Now the algae leaves because it's too hot and try to go into water that are not that hot, because otherwise it cannot survive. You're essentially burning a plant. And it can come back if the eating subside. But if the eating stays for more than a week, 10 days, does not come back, doesn't have the time, that doesn't manage that, okay? So if a bleaching persists for, that's why it's defined by NOAA as the green eating weeks, because really the week period of very high temperature is the problem. So your bleaching goes into mortality if it's more than 8 weeks. You are not going to recover anything at all. And you have very high chance of mortality if you have between 4 and 8 weeks. You can usually recover if the temperature are below 28 degrees for less than 4 weeks. So we have this, and we have also the biodiversity information. And so we can make a recovery index putting those two informations together. And what this map is showing is that if you only know about the bleaching information, okay, so only the temperature and what NOAA tells you, this would be your recovery index. And as you can see, it's good where it's green or it's blue. Blue is better than green. Okay, so the recovery potential is higher where you see blue, is still good where you see green. If you just consider bleaching, this is what you get. So where you don't see any color, the potential of recovery is extremely low. But in fact, it's not the case, because we know that we have a lot of coral reefs in this area that are doing really well. So that was part of the question we were after. If we put together the connectivity and the biodiversity that we get through that connectivity, then we actually match the data. And we do capture the reefs that are indeed recovering much, much better than we expected just from temperature alone. And as you can see, unfortunately, the Great Barrier Reef is always white, no matter what. This region in North Australia is doing much better. It's true. Those are the only reefs in Australia that are still doing okay. The Maldives are doing really well. And in fact, the Maldives are doing really well. Those are reefs that have recovered from the two biggest ENSO events in 3 years, pretty much 100% covered. They lost almost everything. And in 2 years later, they were again working well. Because again, if you bleach, it takes some time before the reef is taken over. So you have a reef that is dead and that is all white, but if you can bring new larvae that can rebuild on top of that reef will start growing again. The problem is that if a reef stays abandoned for too long, then other algae that are really not beneficial to the reef will come and cover it with long algae and that kind of mucus that they think, and at that point there is nothing that the larva can do. So this has been essentially what I was going to tell you today, and the last piece is that I managed to find a paper that looked at the evolution of biodiversity over millions of years. And it's been really interesting to see because, so this is a genetic diversity of benthic foraminifera. So this is reconstruction of diversity based on the fact that when you have high diversity today, you also have many different species of foraminifera, wherever you have it in a marine ecosystem. And foraminifera is another little phytoplankton, okay, that produce calcium carbonate cells. And so when it dies, it falls on the ocean floor, and you can reconstruct distribution of how much you add in different species from pores. So you go in the ocean, you pull up meters and meters and meters of mud, really hundreds of meters of mud, and you have those cores. And then you look at the composition of those core and the amount of delta C13 and the characteristic of those things, and you can find those little shells and reconstruct how much you add and how different they were. Okay, so there has been a reconstruction of biodiversity across millions of years. And 42 to 39 million years ago, most of the biodiversity was in the Mediterranean Sea. Then it starts moving, and by the early Miocene is where you start the coral triangle start showing up. And then today, this is the biodiversity that we have. And so as you can see, it's really high in the coral triangle, and it is the highest, the most diverse hotspot of biodiversity we have in the ocean. And the interesting thing is that here is when we have the first records of henso starting to exist. So really, the developing of that mode of climate variability that didn't exist 40 million years ago, and started to exist around 16 million years ago. It's what built up the biodiversity. So what today we are considering the cause for bleaching a lot of time, in reality is the cause for that hotspot, first and foremost. And also for the fact that that hotspot, that despite the warming, is still managing to survive in some areas, not everywhere, but definitely some areas are doing fine. And we can really map the ones that we know are doing fine. For example, all these areas really doing well. And nobody ever figured out why, because it's been bleached quite significantly. And we can really get a lot of them. So we can also get the ones that are very biodiverse, the biodiversity analysis. So this has been an application of machine learning that we hope we can continue for monitoring large scale. Obviously, then if you really want to protect one specific reef, you have to do higher resolution. But this is essentially mapping you at a resolution of 20 to 30 kilometers, what the ecosystem look like, the marine ecosystem look like for the surface one, so the ophotic. And it can really help in building strategies to protect, because you want to protect the one that are highly biodiverse, for example. You don't want the one that supply a lot of larvae to die. While the one that receive a lot of larvae, you can probably let them die because they're going to recover. So if you have to choose, this is allow you to tell, to say which area you really need. Then you have to choose, because you cannot put protective measurements everywhere. And this becomes very, very important for fishery, because the other thing that you have to do is to limit fishery and to limit overfishing and all the other kind of pollutions for the ones that are really supplying larvae, because we want to maintain them as much as possible. And those are the two people that a lot of the work can represent. And I'm done. Thank you.
I see that we have a nice number of virtual friends. So anyone from the virtual world wants to ask some question? Domenico?
Yeah, I have a question. Yes, I wanted to get some more background on El Niño and La Niña and what they actually do, because I couldn't relate it to the observations of the changes in the connectivity. Perhaps I am not acquainted with these meteorological phenomena in the first place.
Yeah, so El Niño southern oscillation is the major mode of interannual climate variability we have on the planet, and is centered in the Pacific. And what essentially is that if you look at the distribution of sea surface temperature in the Pacific, they are much warmer on the west and cooler on the east. So they are much warmer where essentially the coral triangle is, and much cooler along the coast of South America, all the way into the equator. And the reason for that are multiples. But what happened is that you're essentially piling up waters on the western side, warm waters on the western side, because the winds are continuously pushing water in that direction. So it becomes a pressure and Coriolis system where you have essentially a pile up of water that at some point needs to release. And so all this water moves in the center of the Pacific, fills the whole equatorial Pacific, and you have really a pendulum. So you have a mode of variability that changed that neutral distribution, where you have most of the warm water in the west, spreads the warm water all the way into the east and towards the coast of South America. It's called El Nino because it always peaks at Christmas time. It always peaks in December. And what it does is that by bringing this very warm water towards the coast of Peru and Chile, kills the fishery there on that winter. And so the fishermen didn't like it and start calling it El Nino, and that's how the name came up. And it was discovered in the late 1800s by Walker, Sir Walker, that was sent to India to figure it out why for 2 years or 3 years, for a period of about 10 years, they had a series of very big droughts that caused the tea production that the British really care about to go down. And the reason is that if you have a very strong El Nino in the Pacific, because you are changing so much the distribution of sea surface temperature, and you start changing it, this takes several months, right? Because you have to move all those waters that start usually at the end of spring or December, all the way until they get in December towards the coast of South America and along the equator. And this changed the monsoon system as well, because it's connected. So it changed the water circulation and the air circulation in the atmosphere. And so usually when you have a strong El Nino event, which is where the water really moves toward the coast of South America, you always get a weak monsoon in the summer preceding the peak. And so that's how it was discovered. And because it's an oscillation, and because we have waves, Rossby waves and Kelvin waves that propagate across the ocean, and those are the mechanisms by which you transport this water across the ocean surface. When you get an heat at the coast of South America, they kind of travel back. And so then you pile all this water on the opposite side. So this year, for example, we are in a La Nina year. Last year we had a strong El Nino. And so we hit the coast of South America last winter, and then things start retrieving. The waves moved all this water back. And now we have a La Nina. And then hopefully we will have a few normal years, because it does cause a lot of mess. We are expecting a relatively strong hurricane season, for example, because La Nina changed the winds so much that we tend to have stronger winds in the Atlantic. So usually we do have a lot of hurricanes when it's a La Nina year. Well, last year, despite having very, very warm sea surface temperature over the Atlantic, we didn't have a very active hurricane season, because El Nino completely kills the wind shear, stabilize completely the atmosphere. It's such a big event with these warm waters spreading across the Pacific that the winds, the upper level winds, are essentially dead. And so you have a column of air that is much more uniform. And that really helps in killing the hurricanes. So the hurricanes don't form. So it's the largest mode of variability that we have, and it's really a pendulum, and it's just moving water masses that are very warm, over 28 degrees across the tropical Pacific.
Other questions? Yes.
So I know you mentioned larvae replacing like bleached coral reefs, new corals, sort of revive them. Are humans able to do that same thing in bleached and dead areas? Humans go in and replace the corals?
Yes, we replace corals. We don't replace larvae. Larvae are, so how it happens with larvae and corals, it's kind of an interesting thing. They do spawn once a year only. And they do spawn at a time associated with the full moon in a specific month. So different species of corals have different spawning days, but it's pretty much set. So it can be, depending on where you are, the full moon of August, or a week or 5 days after the full moon of September or November, and depends on the hemisphere and so on. So, and larvae needs to be fertilized. So you have male and female, and they get together, and then they move. And we still don't know how they know when they have the right substrate to attach, and to say, okay, I want to be here. I found my home. We still don't know how that happens. I actually have a student working on that, trying to figure it out if sound is one of the things that makes them figure it out with the substrate. So it's very, very difficult to do that in a lab. Like you can have larvae in a lab, but you're not going to be able to seed larvae. But you can fragment healthy corals and make tiny plants, and then put those tiny plants in the bleach areas, and those tiny plants will grow. And you can grow much faster from tiny segments that you distribute all around than if you're letting a partially bleached plant to regrow itself. So what we do in areas where everything has been badly bleached, especially in Florida, it's to do that. So to grow them for a few weeks in a lab and then have all those tiny fragments replanted. It's very expensive, it's very time consuming. You can do it in very small places. The Red Sea is doing that a lot. The Arabian Sea, huge amount of money poured in trying to save.
Is that helping or is it still dying faster than is being replaced?
Look, I know, I mean, Saudi Arabia is losing the reefs really quickly because it's super hot. I mean, I've seen temperatures that are obscene. They are also trying to genetically modify corals to make them more temperature resistant. So they still have some. I have not seen maps of what the distribution is. In Florida, we lost almost everything. And those efforts have to be very small scale. So you're still not rebuilding, but at least you are keeping, like the Keys corals are rebuilt like that. You're still trying to. It's not sustainable. So there is the idea of engineering corals to make them more heat resistant. In the Great Barrier Reef, for example, they discovered deep corals. So if you go at depth, they have corals. They didn't know they had that many. It's amazing. They have a forest of coral that is 1,000 meters and below. And so they are at least preserving those because the signature of climate change has not been strong enough. So those ones are doing fine. But are different corals and they have different properties.
So the temperature anomalies, they are always skewed. The positive peaks are larger than the negative ones.
Well, yeah, we're warming.
It's always like that.
Well, right now, yes. They're warming. They're
Okay.
definitely warming, and I mean, the signature of the warming, it's quite significant near the surface. Not at all at that.
Okay, so there is a pizza coming in. The last question, yes.
So, thank you. Very interesting presentation, Annalisa.
Can you speak louder?
I have a few questions about the delta maps.
Yeah.
Did you say that you used only the SSTs as the only climatic field to delineate the ecoregions,
Yes.
or did you also use other things?
No, in this case, we just use the SSC's anomalies.
Is there any, I mean, is there any other field that you might add to your accuracy, do you think?
I mean, you can use sea surface height because sea surface heights are directly linked to currents. The problem of the sea surface height is how they are observed. And the fact that in reality, what you observe in sea surface height are rays from satellites that look like this. Those are the satellite passages. And sea surface height is observed like that and then is interpolated by the reanalysis. And unfortunately, when we try with sea surface height, the actual interpolation comes out. So delta map is not happening. It kind of sees the problem. And this is what it gives us currents, which would be the first thing to use. We can use currents from reanalysis, but